Okay, good evening. So today's last session will be given by Dr. Ashok Palniappan on the topic systems medicine. So this will be a lecture session followed by some demonstration. Thank you.
Thank you, Dr. Venkat. Good afternoon, everyone. The topic for the last session today is systems medicine. So I hope you've got a good introduction to networks through today, starting from the morning lecture on networks. So I hope you're able to appreciate the notion of a network and how it can aid a better understanding of biology. And what we will see in this lecture is how the networks concept plays a increasingly important role in unraveling sort of the determinants of disease and how to formulate more effective treatment of disease. Okay, how do we get a better understanding of disease? Okay, so we have lived with diseases since ever since, isn't it? Okay, and we still don't have effective treatments for many diseases, and we don't have a complete understanding of diseases that have been with us for like millennia, like cancer, which is still such a scourge. So there is something that is, that was lacking in our knowledge. Okay, and with the advent of the systems approach and their applications to biology, an important gap in our knowledge is being filled up. And we are able to see that it's not just single molecules and single genes and single proteins that are giving rise to all these pathologies and pathophenotypes, but it's actually a set of interacting players that are working together, working in some pathway, and basically the pathway goes out of gear, and then we have a disease manifestation. So we're not able to exactly pinpoint which is the player that has gone wrong, but what we are able to see is that some pathway has gone wrong, and the pathology results. So this seems like a very a good rationalization of what is really happening. And in order to arrive at this rationalization took us a very long while. And it's truly the network approach to biology that has facilitated this understanding, that it is not just the single genes that we need to be worried about, when we look at a disease, but rather what is the network context of the gene? What are its interacting players? What is its regulatory network? Okay, and any part that goes wrong in this network can affect this particular gene, can in fact affect the pathway outcome itself. And due to this disruption in the pathway, we have the disease manifestation, isn't it? Okay, so this is a new notion of disease, but this notion is what we can really naturally relate to. Okay, not that there is a particular genetic error or something, but rather it's a complex interplay of different factors that is giving rise to most of the common diseases. Okay, so that is the particular approach that we're going to emphasize in today's in this lecture, and the topic is called as systems medicine. The other name for this topic is network medicine. They are interchangeable, systems or network. It's just that we're not looking at single genes and single molecules, but we are looking at them in the context of their neighborhood, in the context of their pathway, where they are actually functioning. None of these are actually working in isolation, isn't it? So therefore, this sort of approach will give us a more integrated, holistic view rather than reducing the system to single elements. Okay, so now we have the technologies and tools and computational techniques to use these approaches. Okay, and therefore we are able to gain better insights into disease mechanisms. All right, so let's go into this a bit more slowly. Okay, so I would just like tuning you into what this systems medicine is. All right, so let's do this a bit more slowly and go over the terms first, and then we will see, what are the methods by which, we can actually gain deeper insight into a given disease. All right. All right. So let's start with this network theory. I know you've been doing this, for the past few hours, but this is, integral to the to what we are going to do, and therefore let's just quickly run over this. So I expect your participation here as well. So let's make this a bit interactive at this point. So elements of network theory. So you've been like listening to all these lectures now. So can anyone tell me what a network is? Can just unmute yourself and can you let me know can you tell us what a network is? Can you define a network?
It is showing the connection and relationship between different elements or nodes.
Okay, very good. Anyone else? So all of you
Sir, a
accept
group of system interconnected with each other through vertices, nodes, edges.
through edges. Okay, okay, excellent. All right, so the first one of you, introduced this term nodes. The second of you introduced this term edges. Okay, so nodes and edges. So this is essentially what a network is. It is just a set of nodes and edges. Okay, so if you give a set of nodes and a set of edges, which are pairwise relations on these nodes, which tell which node is connected to which other node, then you have a network. Okay, whatever may be the size of the set of nodes, whatever may be the size of the set of edges, it may be just one edge and 10 nodes, or it may be like a fully connected network, okay, with 50 edges maybe. Whatever it is, if you have a set of nodes and edges, and these edges are pairwise relations on the nodes, we have a network. So the most general definition of a network is just a set of nodes and edges. Okay, so just keep that in mind. So this network, so this is the network abstraction. That is, we are trying to model something as nodes and edges. So nodes are the players, edges define some sort of a relationship between the nodes, between a pair of nodes. Okay, so it's a set of nodes and edges define some relationships between the nodes. All right, so that's a network. Can anyone tell me what a degree distribution is? Or what the degree of a node is? What is the degree of a node?
Number of nodes connected to each total number of edges connected to each nodes.
Okay.
It's a degree.
Good. Okay, so the degree of a node is the number of edges it has. Okay, so if we find out the degree of every node and if we just list out these degrees, it is called a degree distribution. All right, and this degree distribution will tell us a few things about the network, and you're going to hear more about this tomorrow. So based on the degree distribution, you can say whether the network is scale free or not and all that. Okay, so a degree of a node is just the number of edges, is just the number of edges for a given node. Okay, and the graph can be directed or undirected. So if it's a directed graph, then we can make a distinction between incoming degree and outgoing degree. Okay, so that is the in degree and out degree. So the in degree is the number of incoming edges and the out degree is the number of outgoing edges. All right, so we can make a distinction if it's a directed graph. All right, so that's the degree distribution. And can anyone tell me what a hub is? Have you heard about a hub so far in networks?
Yes, sir.
So what is the network hub?
So, sir, it could be like a central node or like a major important part.
Okay, so a hub is like, like an airport hub. It's the place where most of the flights, have their origin, and you take connecting flights through the hub and all that, isn't it? Okay, so when we say it's a major player, so there are different ways by which we can quantify its importance, isn't it? So one way to quantify the importance, can you think of any way to quantify the importance of a node? A simple way to say whether a node is a hub or not is to look at its degree, isn't it? If the degree is large, then it has a lot of edges. That means it's very well connected with the network. If the degree, on the other hand, is like, less relative to other nodes, then maybe it is not a hub. So one way to find out whether it is a central gene or not, whether it's a hub gene or not, is to look at its degree. There are many other ways. Okay, so these are generally known as centrality measures. Okay, so there are many different centrality measures which will tell us whether a gene is a hub or not. Okay, so knowing whether a gene is a hub or not, we'll see is very important when we come to this analysis of biological networks. Okay, so hubs are a very important concept. And then motifs. We've heard about this term motif in relation to sequence, in relation to structure. Can anyone tell me what a motif means? So what is the motive? All of you are PG students and you've done a lot of, this bioinformatics analysis maybe, and you've certainly come across the term motive. So can anyone define a motive?
So motifs are small regions in a sequence, whether it is protein or nucleotide. It's a protein. In a protein sequence, it's the small regions having three-dimensional structure.
But there may be many small regions that have the, what is characteristic about the motive? Why would one small region be a motive and another region not be a motive?
So, because of the repetition.
Okay. All right. So a motive seems to occur many times. Okay. So that's some sort of a observation. Okay. All right. So anyone else? So a motive, we call some, a particular region as a motive, or a particular sequence, could be a motive if it occurs more often than expected by chance. Okay. So by chance, we have a probability for what we will see. Number of times we will see a particular sequence, but if the particular, stretch occurs more often, or the particular structural, structure occurs more often than expected by chance, then it's called a motive. That means it has been evolutionarily selected. Okay, so it's occurring more often than the average such structure or such sequence. So that becomes a motive. So similarly in networks, you have the network structure, and if a network substructure occurs more often than expected by chance, then that particular substructure becomes a motive for the network. Okay, so how you analyze sequences, how you analyze structures, the same techniques, the same terminology, isn't it? It's going to have a analog even in network theory. Okay, and we can have motives, network motives. Just like we have sequence motives and structure motives, we have network motives. So these are basically network substructures that occur more often than expected by chance. All right. Okay, then we can have modules and clusters. Okay, so we have this motive, which is a very small subnetwork. But if we expand this motive and we get a slightly bigger part, okay, and something like a module, okay, so that's called a network module. It's like a network subnetwork, that is associated with a certain functional trait. Okay, so we have a network and we can cluster the network. Okay, and when we do that, we can, get different subnetworks in the network. Okay, and therefore, if you're recreating or reconstructing a network, we can break it down to different clusters. Okay, so again, so these are all like concepts that are associated with networks. Okay, and these are the concepts that will help us when we want to use this technique to understand diseases better and develop more effective medicines. Okay, motives and then modules and clusters. Okay, so module clusters, these are interchangeable. All right, and then shortest paths. Okay, so what do we mean by shortest path? So we have a network. It consists of nodes and edges. And if you want to travel from one node to the other, there are many different paths by which you can reach the other node. isn't it? Okay, so this goes back to the origins of network or graph theory. Okay, so the other term for network in maths is called graphs, not the XY graph that we usually plot, but as a set of nodes and edges. So that is also called as a graph in network in maths. Okay, so network theory is also called as graph theory, but networks in biology, we can also refer to them as systems. All right. So when we talk about shortest paths, isn't it? So it goes back to the origins of graph theory, like when Euler, the great mathematician, isn't it? Okay, who lived in the 18th century. He basically is like the one who founded this graph theory. And what he did is, the citizens of his area, used to take a short walk every evening, around the town. And this town was actually, connected with a few other islands by bridges. And there were 7 bridges that the citizens can cross and visit all the places and come back to their homes. And there was a problem that the citizens, wanted to visit each of the bridges once and then come back to their home, also visiting all of the places just once. And they wanted to know if they can do this without crossing a bridge 2 times. Just going over the bridge once and then be able to do the entire walk. Is it possible to do that or not? Okay. And they posed this question to Euler. Euler thought about this question and then began to represent the places by nodes and the bridges by edges. And then he began to solve the problem using this node and edge representation. And he came up with the solution that no such walk is possible. Okay, and this is called the 7 bridges of Königsberg problem. And that is really like the founding problem in graph theory. Okay, and so he was like, he came up with this abstraction in the solution of the problem. But this abstraction proved to be very useful and led to an entire field called graph theory. And we're using these same principles of graph theory now, okay, when we want to study, biology and, and pathology in terms of networks and graphs. All right. So the shortest path basically means like, going from one place to the other, in the shortest distance. All right. So when we have this set of nodes and edges, there may be multiple paths between any 2 nodes, but which is the path that yields the smallest distance? That is called the shortest paths. And one assumption we make in biology is whenever we have these 2 players and that are connected by many different paths, they usually communicate with each other using the shortest path. So that is some sort of a working assumption that we use in biology. All right, so these are the working vocabulary for network theory that will be useful to us today as we start with this, systems medicine. All right. So biological networks, I'm sure in the last lecture you heard a full, run through of the many different biological networks. We have a regulatory network. Can anyone tell me what a regulatory network means? What is a regulatory network?
Systems networks that show how a particular thing is regulated.
Regulated, how can it be regulated? What are the ways by which it can be regulated?
Gene regulatory network, maybe either activation or inhibition.
Like there is a feedback system as well. If a particular concentration is high, that can inhibit the other.
Right. Okay. So that is like, we have these enzymes and, these metabolic pathways where you can have these control of the metabolic pathway. So that is one sort of a network. In general, when we speak about regulatory networks, usually we mean genetic regulatory networks. Okay. All of these are regulatory networks. You're very right. And genetic regulatory networks basically relate to, how transcription factors activate or repress other genes, isn't it? So the transcription factor is coded by one gene. So these 2 genes, if the transcription factor of the gene regulates a few other genes, either activates or represses them, then we can put an edge between these 2 genes. Okay. So, and then if we just make a network out of this, it becomes a regulatory network. So that's basically the network of transcription factors. All right. All right, and then we have RNA networks. And how do we actually create this RNA network? We have these non-coding RNA, which have some functional roles. And these are, again, areas of great interest today. And we have these microRNAs, and which were one of the first these functional non-coding RNAs to be found, isn't it? Okay, and now today we have long non-coding RNA, piRNA, all sorts of RNA, non-coding short and long RNAs that we have. Now these RNA basically interact with some other genes, or they interact with some proteins, or they interact with other mRNA of genes, and basically introduce another complex layer of regulation. Okay, so based on the targets of these RNA, we can again create a network. Okay, so if one RNA is regulating another, like mRNA, okay, then you put a edge between these two players, and then likewise, if you create a network, it becomes a RNA network. All right, and then protein-protein interaction networks, which are one of the most easily recognizable networks that we have in biology. So two proteins, this is the undirected network. One protein interacts with another protein. There's no direction to it. And this is like commonly seen in signal transduction pathways, isn't it? In signaling pathways, okay? So, we just take all of these proteins and they interact with each other. We put an edge between them. And in the end, what we get is called a protein-protein interaction network. Such interaction networks are also called as interactomes, okay, interactomes, okay, that is, it is a ohm of all the interactions, okay. So this ohm is a general, 21st century term, or, sort of the systems term that begins to indicate that we have a comprehensive collection of a certain type of information, okay. So interactome refers to an interaction network, okay. All right, and then we have again the metabolic network. As you rightly said, we can have these, enzymes that are linked with each other based on whether the product catalyzed by one enzyme becomes the substrate for the next enzyme. And then we have an enzyme metabolic network. Similarly, we can have a substrate metabolic network, based on whether one substrate is connected with another substrate by an enzyme or not. One enzyme converts the one substrate to another, to the next in the sequence. And of course, they can also be like modulated by metabolites and inhibitors and all these things, which sort of provide a systems control on the dynamics of the metabolic networks. So I must mention here that when we look at biology in terms of these networks, there is one important thing to keep in mind, that these networks are static networks. That is, these are network structures that say that this gene interacts with this gene, or this protein interacts with this protein. And there's no information about what is the strength of the interaction, or what is the rate at which one substrate is converted to another substrate, and all this. Okay, some of those information may be encoded on the edges, but in general, the dynamics of the network is absent during these discussions. Okay, and now that is an active area of research where we want to combine the static representation of networks with the dynamics of the network as well. Now that really goes into the frontiers of research. All right, but as these biological networks are static pictures. They are just pictures of the available information, and they make no mention of the dynamics of any part of this. It's just a static picture. All right. So what I'm going to do now is just go over these organizing principles of system medicine, and we'll come back to these organizing principles towards the end. Okay, after we go through the organizing principles in sequence, and the methods that will help us address each of these organizing principles, they'll make much more sense. So right now, what I want to do is just give you an overview of what we are going to do, and then we'll be coming towards the end and take a check on these organizing principles and see whether we have understood all these organizing principles or not. So the first organizing principle in systems medicine is that a disease phenotype is rarely the consequence of a single gene aberration. Okay, it's not due to some error or mistake in the functioning of a single gene. Okay, but rather it reflects various pathobiological processes collectively that have some aberration going on. Okay, so they interact in a complex network, and there is something wrong that affects the phenotype of the network. It is not just about the single gene alone. So this is the new understanding that we have of a disease. Okay, and this seems like a much more natural definition of a disease. All right. So next is like using network-based approaches. They have these biological and clinical applications from the identification of disease genes to better drug targets. Okay. And we will see how network approaches will help us identify better disease genes. Okay. So that'll, we'll be looking at all this. And then essential genes tend to be associated with hubs, and then disease genes tend to segregate at the network's functional periphery. So this is a principle that has emerged from the study of many disease networks. And what they have found is certain genes that are called as essential genes. We'll come shortly to the idea why some genes are essential and what we mean by essential genes. And it has been found, though, that in a network representation, essential genes tend to coincide with the hubs, whereas the disease genes, a bit counterintuitively, tend to segregate at the periphery. Okay, and we'll see this in detail as well. The next organizing principle is that the disease genes also tend to interact with each other, thus forming modules. Okay, so we just saw that we have these hub genes, and then at a higher level of organization, we have the motifs and the modules. And in fact, it seems that the disease genes, usually are seen together, okay, in a sort of association, okay, and they tend to form these modules, okay, and when they form these modules, we can just call these modules as disease modules. It's like an entire module that is responsible for the disease phenotype. So we have moved from the point where we said that a gene, okay, is the causative factor for the disease. Okay, so initially, like, isn't it 20 years ago, 25 years ago, we would always say that it was this gene, okay, and this gene has a major effect on this disease. So fast forward to today, what we really say is that, this gene is just one player in this network of interacting elements in this pathway, okay, and this gene could have gone wrong, or any other player in this network could have gone wrong. All of these affect the network function, okay, and this network forms a disease module, and any changes, okay, in the genotype, anything, that can affect the resulting phenotype of this network is going to have an impact, okay, on what the disease condition that results, okay. So that's going to result in a disease, okay. So any of the changes in any of these nodes could result in the same disease, okay, and could also contribute to variations in the disease. All right. So this is the new understanding that we have, okay. And I mean, like, given that we have at least 25,000 genes in the human genome, okay, and so it would really be, sort of a stretch to say that one gene or two genes are responsible for a complex disorder like diabetes or hypertension or cardiomyopathy, isn't it? So these are all clearly very complex conditions. And now it begins to make sense that it's not just these one gene. We have discovered these one gene due to some certain experiments and the design of the experiment. But if this gene, we took the entire neighborhood context of this gene, and we were to identify other players in the neighborhood context, and if we were to do the experiment on these genes in the neighborhood, we would again find that these neighborhood genes also are yielding the same disease phenotype, okay? And that seems to be a rational, sort of a view we can adopt, and that has been, that view has been justified. And these views, can be, sort of scientifically pursued in this network paradigm. All right. So next, like, once we are, sort of convinced that there can be disease modules, then what we want to do is we want to find the dependencies between these pathophenotypes. And we know that, the disease modules may overlap between different diseases. Therefore, some diseases may share the same symptoms. And this again accords with everyday experience. We know that headache, for example, or fever accompanies many different conditions. Okay, so it's not like fever is the disease by itself. So fever accompanies many different conditions. Now, why is that? So that is because all these different diseases, they share some common module, and this module that is disturbed in all the diseases is causing fever. Okay, so there is a dependency between the different pathophenotypes, and this can yield something known as a disease zone. So again, a ohm, and this is the ohm of diseases. So what do we mean by ohm of diseases? It's a network of diseases where diseases are linked with each other. That is, diseases are the nodes, and two diseases are linked with each other if they share some common genes, if they share some modules. So therefore, they share some symptoms, for example. So that's the sort of, so now again, from one sort of a valid impact of network theory, we are just taking it forward and applying it at all levels of scientific endeavor. Okay, and now we are at a point where we study diseases as interlinked. Okay, and diseases, we call this as one disease and call that as another disease, but the truth is both these diseases have many symptoms and underlying pathology in common. Okay, so we'll see what are some of the sort of advantages of this approach as well. All right, uh, one thing is it clarifies our understanding of the science. Okay, and from this clarification of the understanding, what is the outcome? There are a lot of applications as well. And so having a true understanding will also automatically, naturally lead to applications, and that is what we are seeing. And okay, all right. So when we find that the diseases are linked at the molecular level, we see that there is a comorbidity between the diseases. That is, if you have hypertension, in due course, you may have a concurrent comorbid heart condition, cardiomyopathy, isn't it? Okay, and if you have diabetes, it seems like your risk for COVID-19 infection is also higher. Okay, so there is always, some comorbidity that is associated. That is, having one disease increases the disposition or risk that you'll have another disease. Okay, so why is there such a comorbidity, isn't it? We have only seen statistical associations between diseases. We did not know why there was this association. Okay, and now we know that significant statistical associations result because diseases share some common nodes in their disease networks, in their disease modules. Okay, and since these nodes are common, since these modules are common, the same symptoms appear in multiple diseases, and also make it more likely that if you have one condition, you may also get another condition. Okay, if you don't treat the one condition, isn't it? So, all right, so that is about comorbidity of diseases. And then finally, this also has implications for drug design. Okay, that is, how do we design better drugs, isn't it? So using a network approach, since we do not know exactly what is the genetic player that is causing the change in the phenotype, isn't it? So it could be anyone in the network that is giving rise to the aberration. So it's not a particular protein that we must try to treat, but it's the network dysfunction that has to be treated. So therefore, when we shift the drug discovery paradigm to treating the network dysfunction rather than a single gene dysfunction, it's an entire shift in the mindset, isn't it? Okay, so this is giving rise to new principles in drug design, and it has given rise to this field called network pharmacology. Okay, and disease classification as well. So what we call as health, what we call as disease. When do you say health becomes a disease? And what is the distinction between different diseases? When you think about it, okay, the more you think about it, the more you will be convinced that all these boundaries are fluid boundaries. Okay, they're all fluid boundaries, and we have to apply these interdisciplinary scientific principles and gain deeper insight into their true nature. All right, so these are the organizing principles. Now we'll again revisit these organizing principles at the end and see how if they make better sense or not. Okay, all right, so let's go into these principles in greater detail. Okay, so disease and essential genes. Okay, so can anyone tell me, what a essential gene means to you? What would a essential gene mean? Anyone?
Sir, is it a gene which causes a function, important function to occur?
Yeah. So what do we mean by important function? I mean, all the genes will have some function. So the idea here is it has some essential function, isn't it? So what do we mean by this essential or important function? How can we define that?
A process that is necessary for cell working.
Cell working, okay. Yeah, all right. Anyone else? Process that is necessary for cell working. Anyone else? But essential, so what is a stronger word to use? Now, is it just cell working?
So metabolism.
Cell metabolism, okay. Anything else?
It is involved in numerous processes.
Numerous processes, okay, good. Anything else?
Which helps in maintaining homeostasis.
Okay. Homeostasis, good. Anything else?
It plays a multiple roles.
Multiple roles. All right, okay. So it's a process that is necessary for, basically, you've said multiple roles, numerous processes, homeostasis, metabolism. So basically, what really comes out of this is that it must be something that is necessary for the survival, okay? Something that is necessary for the survival of the cell, all right? So without this gene, the cell doesn't survive, okay? So knocking this gene out would be lethal to the cell. Then you can say it's an essential gene. So in fact, that is the technical meaning as well. And a essential gene is that, okay, which is without which there would be embryonic lethality. Okay, so we talk about this as a in utero essential gene. Okay, in utero essential gene. So what we mean by that is that without this gene, if we say some gene is essential, that means that without that gene, what we see is embryonic lethality. That is, the embryo doesn't survive. So it is lethal to the embryo if this gene is knocked out or if its function is impaired. Okay, so those are the essential genes. And these essential genes tend to be located in the network hubs. Can anyone think a reason why they should be located in hubs? Why do these essential genes tend to favor network hubs? So suppose we were to draw this network of these genes and then locate the essential genes, we would find more often than not that they are sitting at the hubs of the network. So why do you think that is the case?
Maybe because they're involved in multiple pathways and
Okay, so what do we mean by a hub gene? That is, they are connected with many other players, isn't it? And therefore, if a hub gene is knocked out, then many connections are knocked out. And at least some of these connections have severe effects and which could result in embryonic lethality. So therefore, the network hub seems to be a, sort of a, the natural place for these essential genes, all right? So I hope that is clear. All right, now, compared with the essential genes, there's a distinction to be made. So essential genes are those that are in utero essential, which otherwise would result in embryonic lethality. Okay, so but compared with these essential genes, disease genes are slightly less terrifying. Okay, so that is, they segregate in the functional periphery of the network. Okay, so these are more like, they're not in the hubs, because if it's a hub gene, it becomes an essential gene and that becomes a really serious disease condition. Okay, and which needs to be immediately treated. Okay, and the sort of diseases we are looking at, isn't these are the complex diseases for which most of these genes are located in the functional periphery of the network. Something is happening in the periphery which is easier to happen than to happen in the hub. It's more easy to impair the function of a gene at the periphery than a hub gene, because the hub gene has mechanisms to protect its function, isn't it? Okay, probably there's some redundancy there. But if if this gene is in the periphery, and then it's easy to knock out this gene's function, for example, and these are the sort of, changes, okay, that really, initiate a disease condition, okay? And what we empirically find, so this is the empirical finding, okay? And there may be some new research that has some differences with these or has some new data, and shows some exceptions. But the general trend we observe is that disease genes tend to segregate in the functional periphery of the network. So once we are able to like sort of visualize the network, okay, and see the network topology, based on the topology, we can identify, like using centrality measures, what are the hub genes, okay? And then when we want to identify the disease genes, isn't it? We'll see some techniques to identify disease genes. More often than not, they'll be at the periphery of the network. All right, so this is one way to look at it. And I borrowed this from the founders of systems medicine. Okay, so that is Barabasi and Lokchalzo, and who are both in Northeastern University and Brigham Young University in Massachusetts, in Boston. So so they basically introduced this term systems medicine. All right, and I have used their reviews for some of the figures in this talk. Now, so this is one that highlights the differences between essential genes and disease genes, okay? So let's say we have about 25,000 genes in the human genome, and if we quantify the essential genes and the disease genes and their overlap, okay? So we have about 1,700 essential and 1,700 disease genes, okay? And when we look at their overlap, isn't it? That is essential disease genes, we only get about 400. Okay, so that is only about 25% of the genes are disease genes. It is not that if you're an essential gene, you cannot be a disease gene. That is not the case. It's all statistics. Okay, it's usually that the disease gene tends to segregate at the functional periphery. But there are some disease genes that are also essential genes. And what is the numbers for that? We always go by the numbers. So therefore, number of essential disease genes is 398. Okay, but essential non-disease genes, yeah.
Excuse me, so like the diseases which like affect the metabolism or like the liver function, so those could be classified under essential disease.
I'm not able to hear you. I think your voice broke in the middle, but I think you're asking whether metabolic genes could be classified as essential or disease genes, is it?
No, sir. Actually, what I meant was, say, suppose there is a gene which is affecting the liver function. So those would be coming under essential disease genes, right?
Okay, so that's a good question. Excellent question. So it's affecting the metabolic, the liver function, and therefore it is a disease gene, certainly. Okay, it fits the definition of a disease gene. But does it fit the definition of an essential gene? We do not know. Okay, so on a gene,
Sir,
on a gene by gene basis, to be an essential gene, it has to be embryonically lethal.
Sir, but like another question, sir, follow up. So it's like, say, suppose like the Gilbert syndrome is there. So that actually causes the person to have a higher concentration of bilirubin always, like constantly. So, but that is not a harmful thing. Like it is like, it is kind of genetic. So, how, like, what is your comment on this, sir?
Yeah, so those are sort of what we can call as Mendelian disorders, which are single gene disorders that have an inheritance pattern as well. So that's an excellent question too. Again, so, so this network view, the systems view is usually applied to these complex diseases. When we have the Mendelian diseases, it's very clear what the causative factor is. It's usually a single gene or a couple of genes. And we know exactly what mutations in this gene is going to cause the Mendelian condition, okay? So what we have is the Mendelian condition, and it is related to a single gene. It's a monogenic disorder, okay? So we do have monogenic disorders, which might be modulated by the network context, but are largely independent of the network context, okay? So the network context is the natural context, but there are some conditions that are given rise to, okay? So everything we say does have an exception, isn't it? So there are conditions that are given rise to by a monogenic condition, okay? So for example, sickle cell anemia is due to a point mutation that results in the sickling of the red blood cells, isn't it? So there, whatever may be the network context, because the hemoglobin has this, aggregation property, which will deform the red blood cells and eventually resulting in sickle cell anemia. Largely independent of network context, you're going to have the condition. That is, you're going to suffer from sickle cell anemia disease if you have that particular SNP in the hemoglobin gene. All right. So this is a Mendelian trait. Okay. So biology is very complex, isn't it? Okay. So networks are one way to to clear this confusion. But we have to keep in mind, that nature can be truly complex. All right. So that is, I hope that answers your question. Okay. All right. So that is about, the second visualization here is that, let's say, this is a particular network, isn't it? And this network is like engaged in some process. And what we find is the nodes that are central, here just by degree centrality we are saying, and that have high degrees, they can be the hubs in the node, and they usually tend to correspond to essential proteins. But those that are at the periphery of the network, they are the ones that are likely to be involved in disease processes, because these are the ones that are easier to go wrong. Okay, and they are the ones that give rise to all the common diseases that we see, the chronic conditions. So this is like a new understanding of these conditions. All right, so moving from disease genes to disease modules, okay, network modules. So let's first talk about what we mean by a module. So there are a few types of modules that we can define. First is a topological module, which is from purely a mathematical point of view. If we have a network, what is a module? So that is based on the structure of the network. That is the topology of the network. And wherever we find a locally dense neighborhood, we can say it is a module, okay? And within the module, we find that the nodes of the module have a higher tendency to interact with each other than with nodes that are outside. So that becomes a contained module, okay? And it's a pure network property. Next, we can define some functional modules where we are going from mathematics into biology, okay? So where we have these topological modules, and those topological modules that can also be associated with a biological function, okay? So if I have a biological function and I can map the biological function onto this module, okay? So if you're familiar with the gene ontology, okay, so we have a molecular function that is mapped with every gene. We also have a biological process of which the gene is a part of, and this biological process consists of many other genes as well. So that is one sort of a functional module, isn't it? So if we have a topological module that does map into these biological processes, then what we have is a functional module. So it's a network neighborhood with with nodes that are all related in function. Okay, so this is a nodal characteristic. It is not a network property alone. Now we are also adding to it a condition that it must be associated with a biological function or a biological process. So it's a nodal characteristic, and the nodes involved in closely related cellular functions tend to interact with each other more. And they're also showing tendencies to be located in the same network neighborhood. And these are called as functional modules. Now moving from biology into medicine, so we have topological modules, a mathematical construct, then we have functional modules, which is a biological concept. Now moving into medicine, what we have is a disease module, okay? Now what are these disease modules? They are basically, again, a neighborhood or a group of nodes. But if you were to apply a perturbation to this group or to any node in this group, it can be linked to a particular disease phenotype. Then that is known as those functional modules are also called as disease modules, okay? And these perturbations can be anything, they can be like mutations or deletion, genetic deletions, or like, a copy number variation or some expression changes or some alteration in the protein function due to the mutation or some post-translational modification, okay? Whatever might alter the function of the nodes in this particular neighborhood. So those are called as disease modules. Now, what are these? So this is a graphical view of the same. So if you had this network from a purely graph theoretic point of view, this becomes a topological module. And then what is the part of this module that can map into a biological function? It may not correlate exactly with the topological module, but for the same network, this may be the functional module. Now, even given this functional module might not correspond exactly with the disease module, because to correspond with the disease module, some perturbation to the nodes should yield a particular disease phenotype, and that may result in this being the disease module. Okay, those in orange color here. All right, so this is a topological module that is marked here, and for the same network, this is the functional module, and for the same network, this may be the disease module. All right, and all the genes in the disease module basically are disease genes. All right, so all right, so functional modules have to overlap with these topological modules. So now we see a new concept of a disease that emerges from these different notions of a module. So what is the disease? Now we can say a disease in a network term. Okay, that is, a disease can be defined in terms of a disease module. That is in terms of a functional module. If there's a breakdown in the function, then it's going to yield a disease phenotype. So any breakdown in functional modules which corresponds with the disease module, okay, they are basically the diseases that we are familiar with. So a disease can be viewed as the breakdown as a functional module. Okay, and this is a new understanding that we have from a network perspective. All right. All right, so now we have seen about what are hubs and modules in the context of systems medicine. Okay, so we started with a network concept, then we moved on to a biological concept. Now we have moved into a medical network medicine concept itself, and we know what these hub genes, disease genes are, and what these disease modules are. Now that the concepts are clear, let us see how we can actually detect disease genes. And how do we detect disease modules? What are some methods that will let us do that? All right. So methods to identify disease gene candidates, can be broadly grouped under 3 different methods. First is called the linkage method, where how do we identify a new disease gene? Okay. So when we do the linkage analysis, we know a certain interval in the genome that is correlated with the disease transmission. Okay. So now, if in this interval we find some genes. Okay, and if these genes also happen to interact with known disease-associated proteins, then we can apply the network concept, and these are in the interacting network and in the same linkage interval of the disease. So maybe these are also disease candidates. All right, so that is one way. The second way is we can use the disease module concept. Okay, so we have reconstructed a particular network, and then we cluster the network or partition the network into different modules. And where these modules contain known disease-associated proteins, then the interacting genes in that particular module also become candidates for disease genes. All right, so that is a module-based method. So 2 methods that use network knowledge. And third is diffusion-based method. Again, we start with a set of few genes that we already know to be associated with the disease, and then we reconstruct the network. And then what we do is we use this sort of a random walk theory, okay, which is a mathematical simulation principle, where we have a walker that randomly chooses, that starts from some node randomly and chooses to visit other nodes in the network with a certain probability. We assign a probability to visiting all the other nodes, and then it samples all the nodes in the network for a certain length of time, for a length of simulation time. At the end of it, we know how many times it has visited each node. Now, based on, if it were to start at the disease nodes, known disease nodes, then we really find what is the other nodes in the network that have close affinities with the disease node. So that is called a diffusion-based method. Where we start a random walker on known disease genes, and it samples all the other genes in the network, and based on how many times it has visited the other nodes, we can rank the other nodes in the network and then say these have the high probability to be the other candidate disease genes. Okay, so we get likelihoods that for all the other nodes in the particular network. So this is a visualization of that, again from Barabazi and Lokchalzo, with Gulbachi. So here the linkage method. So this is a disease linkage interval, and these are the known proteins associated with the disease. And in this linkage interval, we find 3 genes, and 2 of these genes interact with the known proteins. One gene has no known protein-protein interaction with a known disease protein gene. All right, so we can say P1 and PN now are good disease gene candidates. Similarly, here we have identified a disease module. We'll see how to identify disease modules separately shortly. All right, so once you've identified a disease module, and it's known that the blue ones are the disease-associated proteins, and what are the other genes that are talking with these disease-associated proteins in the disease module, those are the candidate proteins. So now we can really expand our, sort of a set of targets for the given disease condition. Okay, so in addition to yielding some insights into the physiology of this condition, what we also have is some new biomarker or new target for the particular condition. All right, and this is again a diffusion-based method. Okay, and it starts at some known protein, disease-associated protein, and visits all the other genes, and then we get a likelihood for whether each of these nodes could also be a candidate disease genes. All right. Now coming to disease modules. Now, how do we identify, what are the methods for disease modules? So we have seen how to identify disease genes, a few methods. Now, how do we identify disease modules? And here, it's a sort of series of steps that we do to identify the disease modules. Within each step, there are many different ways to do the steps. But we have to go through this set of 4 steps to identify the disease module. So the first sort of a step is to reconstruct the intractome, whatever disease your is of interest in whatever tissue and cell line. For that particular tissue or cell line, reconstruct the intractome. Okay, and to reconstruct the intractome, we will use all the network information we already discussed. That is the regulatory networks, the RNA networks, the metabolic network, protein-protein interactions. So all these can go into recreating this intractome network. And this can also be augmented with co-expression based relationships. That is, we use expression data, see which genes are co-expressed, and then maybe their similar expression profiles indicate that they're also interacting or involved in the same pathway, isn't it? All right, so these are all principles from other areas that we can apply here and then reconstruct the intractome. So the first step is to reconstruct the OME, that is the network. Okay, we want to get the full picture of the network. Next, from this network, in this network, identify the known disease-associated genes. So we're not starting with a blank slate. We already know there are certain genes that are associated with the disease. What we want to do is identify all the genes that could be associated with the disease. But we have a current knowledge, okay, from so much medical work, isn't it? That there are certain genes that are already known to be associated with your disease of interest. And this could be obtained from linkage analysis or GWAS and other sources. So use these, okay, and which constitute the seed of the disease model, which constitute the seed set of genes for the network. Okay, so first reconstruct the intractome. Second, identify the seed set of genes. Okay, so in the intractome, okay, identify all the genes that are the seed set for the given disease. Okay, and once we have the seed set, the next step is to identify disease modules. Okay, from the seed step, we've already seen how to identify the modules. That is what we discussed in this previous slide. Okay, so you have the seed set of genes P1, P2, P3. From that, we identify P4. Or we have these 2 genes here, and we use a diffusion-based method and identify the other genes. So from the seed set, we can identify all the other disease genes. Okay, so reconstruct the intractome, seed the network with the known disease genes. From there, identify the disease module. So this module is the one that contains most of the disease-associated components, most of the disease seed genes that we obtain from the reconstruction. Okay, and if this network shows statistically significant agglomeration, okay, this can be detected using clustering tools. That means it's a disease module that is clearly associated with the disease. Okay, so this is like a functionally and topologically compact subgraph. Okay, that can be directly mapped with the disease status. Okay, so this represents the potential disease module. Okay, so in this way, we can identify the disease module. Okay, so there's a very clear workflow here. And from these disease modules, sometimes what we find is that the disease module itself is very large. Okay, and if the disease module is very large, then it's not very useful in terms of applications. It's not tractable for downstream analysis. So what we then need to do is, from this disease module, we need to seek molecular pathways, okay, whose disruption would specifically result in the disease phenotype. Okay, so from the disease module, what we have to do is use shortest paths between nodes that are known to be associated with the disease, and take all the players nodes in the shortest path and say that constitutes a molecular pathway, and we say that this molecular pathway now offers us a sort of a network view of the disease, something that we can use for downstream applications. So we use the network parsimony principle to select the most likely disease pathways. By parsimony principle, we mean that when we have two nodes, they talk with each other using the shortest path. Okay, parsimony means like some economy, okay? And so when two nodes need to talk with each other, they use the shortest distance between them. So that is the parsimony principle. So those indicate the actual likely biological disease pathways. All right, so just to recap this, the steps in this: first, we reconstruct the interactome, then we identify the disease, we just seed the interactome with the known disease genes from many different sources. And then using the seed genes that are those in red color, those in blue color, we can identify the disease modules and the candidate disease genes that are part of the module that is identified by the seed set of genes. And if this is one module, we can break it down into these two subnetworks as well. And you see that there are some overlapping proteins as well, but these could be the pathways. If the disease module is too large, we can break it down into separate pathways in this manner, and then begin to target one of these pathways for the downstream applications. Okay, so these are the pathways, and we can validate whatever we have identified here. So these are all like computational analysis and omic data analysis. From this, we again have to validate it, and we can do this using multiple techniques. All right. Okay. So now, I hope this has given you a very nice overview of, the why we have to use a network approach, isn't it? What is indispensable about the network approach? I mean, if you don't use the network approach, what sort of information we won't be able to gain about a disease? Okay. So the network approach, let's say, is like essential to a deeper understanding of diseases. Because it is true that genes and proteins and all the biomolecules are working in a context, are working in a neighborhood, isn't it? And therefore, any disturbance and aberration that we see, does not stem, exclusively from a single gene, but it could be a neighborhood effect or a context effect, isn't it? And it could be, shortly, in other words, it could be a network effect. So we have to use this network approach. And, I hope this has given you a very nice overview. So that is the crux of the talk, okay? And what I want to do now is, so once, we use this network approach, and then we can identify candidate disease genes, we can identify disease modules that actually tell us exactly what is going on in the disease. Then what we can do is we can go and examine comorbidity between diseases. Okay, so this was a separate study that was carried out, and what they did is they took all the diseases that were known, and then diseases were the nodes, and they joined two diseases with each other if they shared some nodes. Okay, so the disease itself can be given as a disease module, and let's take two disease conditions here, and one is breast cancer and the other is bone and cartilage cancer. Both are cancers, and if you see the disease module for the breast cancer shares the couple of genes with the disease module for the bone and cartilage cancer. So therefore, these two diseases will be linked. So that is how we define an edge. So the nodes are all the diseases. An edge represents a linkage between two diseases if they share some common genes in their disease module. In this way, an entire research study was done, and what they did is they obtained the network of all diseases. They obtained the mapping of the universe of diseases, that afflict human, isn't it? Okay, so which is a very fascinating study, and this was, goes back to 2007. Okay, so 2005 was the time when this notion of systems medicine was introduced, and we had the human disease network. Okay, and so this is the disease network, and we see a cluster here of green color, and most of these are cancers, different cancers. Okay, and you see something here. You have this one is diabetes, and you see it's the size of the circle, tells us how many other diseases it is connected to, and you have diabetes, which is a visible circle, and then you have some hypertension here, and then you have cardiomyopathy somewhere here. All right, so this is a very interesting study, and those of you who are interested in knowing about, what are the what could be a taxonomy of diseases. So this is a network taxonomy of diseases. All right. Okay, so that really, and there are many more directions in which we can go, applying this network idea, to many different sorts of data and coming up with our own networks and analyzing these networks using known tools like centrality analysis and cluster analysis, and interpreting the results of these different analysis, which can yield insights and really advance, our sort of efforts to treat a lot of disease conditions. All right. So what are the hypotheses? So towards the end, what I want to do is again run over the hypothesis. Now that we have gone over this sort of a introduction to network medicine, there are some assumptions we have been using throughout. And now what I want to do is spell out what these assumptions are, that have been implicit in our discussion. So one is hubs. So there exist some hubs, and these hubs usually tend to be in utero essential genes, whereas disease genes tend to avoid hubs. Okay. So with respect to networks and their hubs, network medicine tells us that disease genes tend to avoid hubs. They only tend to avoid, but there are quite a few disease genes that are also hub genes. All right. Okay, next is the local hypothesis. That is, the proteins involved in the same disease tend to interact with each other, and that is, they are in a network neighborhood. Okay, and the corollary of the hypothesis is that mutations in these interacting proteins usually lead to the similar disease phenotypes because they are affecting the pathway, and the pathway has a certain biological function, and it is that biological function that is affected. Due to that disturbance in the biological function, the phenotype is usually going to be similar. Okay, so even though the causes are different, okay, the disease phenotype is similar, and this is the key point we have to pay attention to. All right. Next is the disease module hypothesis. That is what we say is that the cellular components associated with the disease phenotype show a tendency to cluster in the same network neighborhood. Okay, so this network concept that we have been using is what facilitates, okay, to identify these disease modules. Next is the parsimony principle, where we assume that the causal molecular pathways usually coincide with the shortest molecular paths between the nodes that are disease associated genes. All right. And finally, the last hypothesis that is implicitly assumed in our discussions is the shared components hypothesis. That is, diseases that share certain components show phenotypic similarity and may also be comorbid. Okay, so that is known as the shared components. That is the disease zone that we see here. So where two different conditions share a few genes, and therefore they show some similarities. Both of these are cancers, and there is a set of hallmarks for all cancers, and that is exhibited by all the cancers. That may be one of the reasons why we find some of these players, in any cancer condition, tend to overlap with each other. All right. Okay. So this is some new research, just to sort of bring our discussions up to date. So this was just published, I think, in 2020, less than a year ago, and here you see that this network approach is now recast into a neural network. I'm sure all of you have seen a neural network. So there are some input layer, output layers, and there are some hidden layers here. And this entire network discussion we've been having about genes that lead to pathways that lead to diseases can be given this sort of a neural network architecture itself. And there was a study that tried to cast the genes as the input layers and disease as the output layers, mediated by the biological pathways in the hidden layer, and they were very successful. Okay, and you can look up this reference for more details on that. And finally, I want to touch upon what the situation that is leading us, led us to this virtual workshop as well. That is the COVID-19 pandemic, and we were to apply this network approaches, and what is the how do we do that, and what is the sort of the outcome of that. and what are the uses of that, and is it helping us or not? So those approaches, generally come to be called as systems pharmacology. So one is medicine, and pharmacology is a subset of medicine, where what we are interested in is the discovery of new drugs and the action of the drugs, okay, towards the treatment of the disease. So this sort of a systems medicine is more specifically called as systems pharmacology. And here what we see in A is the pathogenesis of the SARS-CoV-2 virus, the coronavirus, new coronavirus 2. And then in order to obtain the disease module, okay, for the SARS-CoV-2, what we do is we take the proteins in the SARS-CoV-2 and identify the human target genes and the target proteins from the transcriptome and proteome, human transcriptome and proteome, as well as the, and then we use the interactome information and expand the network, okay? So we have to generate the host target network from the COVID-2 proteins. And once this network, this interactome is reconstructed, and it can be validated with the COVID-19 registry, this becomes the COVID-19 network. Now what we can do is use this COVID-19 network and compare it with the known networks for the other diseases. Already I told you that there's been a study that mapped all the diseases together, and therefore we know for every disease what the disease module is. So now that we know the disease module for COVID-19 that we obtained from this, using the host target genes and proteins, okay, so the COVID-19 network again can be compared and mapped with the known networks for the other conditions. And what we find is that there are some overlapping disease modules between the coronavirus 2 and some existing conditions, okay? So why does COVID-2 lead to some pulmonary conditions, some metabolic conditions, some, even increase a risk for cancer? Because there are some shared components between the coronavirus 2 network and the cancer network, between the coronavirus network and the metabolic network, okay? And that's the outcome of the study. Now, based on all these, they've also done some multi-omics visualization, and some of the drugs that are useful in these conditions can also be used against the coronavirus 2 and can be validated. And then if it's effective, these repurposed drugs can be effective against the COVID-19. So this is a very new work that has just been published, and it's gained a lot of media attention as well. And this shows like a classic application of all the network literature we have so far, and the network techniques that we have for this emerging for this pandemic situation we're all going through. Okay, and so this is like very relevant, contemporary, and gives us probably a wider picture of what we're going through with respect to the mutations in the COVID-19 and everything. How do we? So what are some ways by which we can have some robust treatment of this condition? All right. So final thoughts. One is with respect to the network pharmacology that we just saw. So single target drugs will only correct some dysfunctional aspects of the disease module. Okay. So mutation in one protein can affect the disease module. Mutation in another protein can also affect the disease module in the same way. If we are treating the mutation in only one of the proteins, then the other protein, if it's mutated, it is untreated, isn't it? So therefore, single target drugs may correct some aspects, but not all aspects. Clearly, what we need is a polypharmacology. Okay. So moreover, single target drugs may also alter the activity of molecules that are situated in the neighborhood of this protein, and this leads to side effects. These are unintended effects, and these are the side effects which we are all familiar with. We take some medicine, and that medicine works for someone, but it doesn't work for us, and we have some side effects. It's a common everyday experience, isn't it? So that is because, okay, so the symptoms are the same, but personalized way, what is causing, for us, the causative factor is slightly different from the causative factor for someone else. So even though we take the same medicine, it only causes side effects. It also causes side effects, isn't it? Okay, the solution is polypharmacology, where we use drug combinations. And the problem here is to identify what is the optimal drug combination, isn't it? Okay, and some applications of this we have seen in AIDS treatment, where the standard now is to use a cocktail therapy of 3 different drugs. It's not just one drug, but 3 different drugs. And this is the treatment that has now proved to be effective. All right. Now, moreover, we can also go ahead and construct disease drug networks, drug similarity networks, so many different networks from the sort of data that we have here. All right. Another, sort of a big outcome of all this network research is that we're getting a better idea of disease classification. You may wonder, what is there to know about diseases? What is there? What is new to know? There's a whole lot. Now, I hope this particular discussion has convinced you that we only know some things about diseases. Okay, what is really happening in a network manner, isn't it? And what are all, how do we better define a disease, and what is the difference between one disease and another disease, and why do we share symptoms between some diseases? Okay, and what is the distinction between health and disease? Okay, so there's an interconnected nature of many diseases, and therefore we need to define disease susceptibility, disease outcome. We need to identify tailored therapeutic strategies. So this sort of a network approach is indispensable to provide a framework for all these different things. All right, so the last thing I want to talk to you about is having a practical orientation to this. So we have seen the methods and we have discussed the methods. I just want to spend 1 or 2 minutes on how do we do this in a computational way. So there's a software for this called a Cytoscape for doing biological network analysis, developed by Idecker's lab in University of California, San Diego. And it has been widely used for the last 15 or so years for all sorts of biological network analysis. So Cytoscape is a general Java-based platform. And the good thing about Cytoscape is that we can have plugins for Cytoscape to do very specific analysis. In this way, Cytoscape has a very active community of that develops plugins and keeps increasing the database of plugins that are repository of plugins that are available in Cytoscape. Okay, and we'll see how to do some of the analysis in Cytoscape using these plugins. That is, I'll just like let you just indicate what these plugins are. So to do the network analysis, we start with a seed set of genes. These could be obtained from experiments, GWAS, whatever source that we have discussed so far, even from computational analysis. From these, we have to reconstruct the entire network. And how do we do that? There are some plugins for this. Okay, so the seed set interacts with a lot of other genes. And like this, we can keep expanding the network, isn't it? Okay. And some plugins are Gene Mania and Reactome that does this network reconstruction. From this network reconstruction, we have to identify maybe hub genes that will let us know, what is the module structure. And for this, we can use many different centrality measures like degree centrality, betweenness, closeness, eigen. All these measure slightly different notions of what it means to be a central gene. And there are many different plugins to do this, like Centrality, NCA, Centiscape, and all that. Now, once we have these, part complete, then we can decompose the network into the modules. For this, we can use clustering or affinity propagation, all these different techniques. And these techniques are implemented in many different Cytoscape plugins, like Cluster Maker, Moduland, and all that. And then we can get some disease modules, and some of these genes within these modules would be candidate disease genes. All right. So there is a vast ecosystem of plugins that you can further use and analyze the networks that have been reconstructed. And so the network reconstruction is the key part. All right, and then you can further, once we have these disease modules, we can find out what pathways are associated with the disease using plugins like Bingo. And then you can knock out a gene in the network and then find out what is the effect on the network. So many different, you can examine in silico essentiality of a gene as well, so many different things can be done in Cytoscape. Okay, so, it would take, a full lab course to do that. All right, so I hope this has given you some sort of an overview of systems medicine, So one of the main objectives is to impart what it means to have a systems view, okay? And why the systems view is essential to a accurate understanding of disease, okay? And then I hope we have been able to understand what it means by what is meant by a disease gene, okay? And how we can identify the candidate disease genes. And moreover, what is meant by a disease module that actually represents the disease network or the disease neighborhood, and how do we identify the disease module, okay? And I hope you're left with the appreciation of the applications of this network approach. All right, so we have just like a few minutes for questions. So if anyone has any questions, please go ahead. Any questions, anyone?
I have a question.
Yes.
So out of curiosity, I wanted to know whether the concept of network can be mapped to study the allostery within a protein?
Absolutely. Okay. So what it's an excellent question. Thanks for asking that. Okay. So this is just the question that you asked basically leads to another application of networks. Okay. See, the only limitation is imagination. Where can we apply this network concept? Okay. We have seen networks in the context of regulatory networks and the interaction networks and metabolic networks. But then let's think about the residues in a protein. Okay. And we have let's say we have all these many residues in the protein. And then what we have is one residue, contacts another residue if they are
Yes.
close enough to each other. So now what we say is we say that two residues are connected with each other. Okay,
Yeah.
that is they share an edge if their distance is less than some certain
Cut off.
threshold.
Cut off.
Okay, so if at least one atom of this residue is within this threshold of at least one atom of that residue, we say that two residues are linked. Now, in this way, you can generate a residue interaction network. Okay, and these are called RINs, residue interaction networks. And then once you create this network, you can do a structural analysis of this network. Okay, and then if you have some allostery in the protein, isn't it? There is a part of this network that is involved in the allostery. So go ahead, apply these network techniques we just talked about, and I'm sure you can study the allostery.
Okay, so actually I was looking for a methodology it was out of curiosity. I wanted to know whether it is possible, because here the genes are, we do even, you say that there is even possibility to study we can do a gene knockout and study the effect. So likewise, a similar kind of approach can, whether these kind of studies already exist or for allostery.
For allostery, I'm not sure, so I mean, there's just so much literature out there, so I'm not really sure, but I think it's a very interesting question and it's worth pursuing.
Okay, thank you, sir. So,
any other question, anyone? So that's a very interesting question, that is a new type of network you can create. All you have to do is, define what the nodes are and define what constitutes an edge. So as long as you can define what constitutes an edge, now you have a network. And then once you have a network, we can bring to bear the existing techniques, that are there to analyze networks onto the topic or problem we have at hand. So we have done in our lab, in my lab, we have done, we have done some work on cancer biology. We have identified cancer hub genes. Okay, so there it's actually like key genes that drive the cancer, and then we have identified the disease modules there and identified some potential new genes that could not be uncovered from the experimental data, but using a network approach, we are able to identify some potential new genes that are involved in cancer processes. Okay. So those sort of
Thank you, sir.
Any other questions?
Sir, about the cancer biology study which you just mentioned right now, so is there a literature which we can actually peruse for our own curiosity?
Yeah. There's, I mean, a lot of the computational cancer studies use network methods. The one we have done is a publication in PLOS One, and let's see that BMC cancer. So I think 2016 PLOS One, we have a computational identification of disease biomarkers in colon cancer. And then in 2018, I think we published on state-specific biomarkers in hepatocellular carcinoma. So both these use omics data, and from the omics data, we reconstruct a network, and from the network, we have some more analysis as well. And we do a lot of other things as well. So, but it's a part of the study. Okay, so the main idea that I want to really convey to you all is that as long as we can have some nodes and a definition for an edge, you have a network. And once you have a network, we can use the 300-year-old graph theory of mathematics to bring to bear on the problem. What started with Euler, those techniques today, and we have the software for these techniques as well. And it's a very active area in maths as well, this graph theory, and all of those, knowledge, interdisciplinary knowledge can be brought to bear on these problems. And the field just grows. And most of the people who advance the field are in fact physicists. Barabasi, whom I mentioned as one of the founders of the systems, that is who initiated this concept of systems medicine. Now he is a physicist too. So.
Sir, is there any other way to determine a dominant node in a complex network other than centrality measure?
All right. See, when we talk about dominant node, we may mean a node that has high degree, or we may mean a node, if you take out the node, that means information flow in the network is the most heavily impaired, okay? Because the path lengths begin to increase and all that. Or the node may be important due to being a eigen node for the network, that has to do with a translation into linear algebra, matrix format and all that. So there are many different ways by which we can quantify the importance. Now, all these important measures are generally called under the umbrella term centrality measures. And there are many different centrality measures. Okay, I just told you like 3 or 4, but there are some 30 to 50. If you look in this plugin called Centiscape, it has a catalog of all these centrality measures. And there are some 30 or 40 centrality measures it implements. And I'm sure one of the measures would correspond with the idea that you have. You have some idea, some notion of what is a dominant node. I'm sure that idea is represented in one of these centrality measures. There are just so many of these. There is like eccentric centrality, stress centrality, MCC centrality. I don't know. Lot of different centralities are there. And whatever definition you may have for your dominant node, that becomes another centrality measure. Basically, if it's not there already, then it becomes a new centrality measure. Again, it's called as a centrality measure though. So yeah, so thank you all. So I hope that was informative to all of you. If you have any questions, feel free to write to me. This is my email address. So right here. And so thank you all.
Okay. Thank you, Dr. Ashok, for the talk. That concludes today's program. We'll see you again tomorrow morning 9 a.m. Thank you.